Considerations regarding the sweet. Risk. A compound substance in typical sugar influences DNA. Introduction. Sugar has long been a staple in diets across the globe, often celebrated for its sweetness and versatility. However, emerging research has begun to unravel a more complex narrative regarding the consumption of sugar, particularly its impact on our DNA. This article aims to explore the various dimensions of sugar consumption, focusing on a specific compound substance found in typical sugar and its potential influence on DNA. Understanding sugar. Sugar, or sucrose, is a disaccharide composed of glucose and fructose. It is found naturally in many fruits and vegetables, but is also added to numerous processed foods. The human body metabolizes sugar to produce energy, making it a critical component of our diet. However, its excessive consumption has been linked to numerous health issues, including obesity, diabetes, and heart disease. The structure of sugar. Sucrose is composed of two monosaccharides, glucose and fructose. Glucose is vital for energy production in cells, while fructose is primarily metabolized in the liver. This structural composition is significant because it influences how our bodies process sugar and its subsequent effects on health. Types of sugar. There are several types of sugar, including table sugar, sucrose, high fructose corn syrup, and naturally occurring sugars found in fruits, fructose, and dairy, lactose. Each type has unique metabolic pathways and health implications, particularly concerning their impact on DNA. The role of DNA. Deoxyribonucleic acid, DNA, is the hereditary material in all living organisms. It carries the genetic instructions for the development, functioning, growth, and reproduction of all known organisms. Understanding how various substances interact with DNA is crucial for comprehending their broader health implications. Glycation, a hidden risk. One of the main concerns with sugar consumption is glycation, a process where sugars bind to proteins or lipids without the controlling action of an enzyme. This interaction forms advanced glycation end products, AGEs, which can contribute to cellular damage, inflammation, and increased oxidative stress, potentially affecting DNA integrity. Sugar and oxidative stress. Oxidative stress occurs when there is an imbalance between free radicals and antioxidants in the body. High sugar intake has been shown to increase oxidative stress, which can damage cellular components, including DNA. This relationship underscores the need to understand the impact of sugar on genetic material. The epigenetic effects of sugar. Epigenetics refers to changes in gene expression that do not involve alterations to the underlying DNA sequence. Research indicates that high sugar diets may lead to epigenetic modifications, impacting gene expression related to metabolism, inflammation, and disease susceptibility. Sugar and cancer risk. There is a growing body of evidence linking excessive sugar consumption to an increased risk of certain cancers. Elevated insulin levels due to high sugar intake can promote the growth of cancer cells. Additionally, the glycation process may play a role in tumorigenesis by promoting oxidative stress and inflammation. The impact on gut microbiota. The human gut microbiota is influenced by dietary habits, including sugar consumption. High sugar diets can lead to dysbiosis, an imbalance in gut bacteria, which may indirectly affect DNA health. A healthy microbiome supports immune function and DNA repair mechanisms. Inflammation and sugar. Chronic inflammation is a risk factor for many diseases, including those that affect DNA integrity. High sugar intake has been shown to promote inflammation through various mechanisms, including the release of pro-inflammatory cytokines and the production of AGEs. Sugar's role in metabolic syndrome. Metabolic syndrome encompasses a cluster of conditions that increase the risk of heart disease, stroke, and diabetes. High sugar consumption is a significant contributor to this syndrome, potentially leading to alterations in DNA that predispose individuals to these conditions. Cellular repair mechanisms. The body has robust mechanisms to repair damaged DNA, but these systems can be overwhelmed by excessive oxidative stress and inflammation caused by high sugar intake. Understanding how sugar influences these repair pathways is crucial for mitigating its risks. The influence of artificial sweeteners. While artificial sweeteners are often marketed as healthier alternatives to sugar, their long-term effects on DNA and overall health remain contentious. Some studies suggest that they may disrupt gut microbiota, potentially leading to negative health outcomes. Sugar and processed foods. Processed foods are a major source of added sugars in the modern diet. Understanding the impact of these foods on health and DNA is essential, as they often contain not only sugar but also other harmful additives that may exacerbate health risks. The role of education in sugar consumption. Public awareness regarding the risks associated with excessive sugar consumption is vital. Educational campaigns focusing on the effects of sugar on health, particularly its impact on DNA, can help individuals make informed dietary choices. Future research directions. As research into the effects of sugar on DNA continues to evolve, future studies should focus on elucidating the mechanisms by which sugar influences genetic material. Longitudinal studies examining the impact of sugar on epigenetic changes and disease susceptibility will be particularly valuable. Conclusion: the relationship between sugar consumption and its impact on DNA is a complex and evolving field of study. While sugar remains a significant part of many diets, awareness of its potential risks, particularly regarding DNA integrity and health, is essential. By understanding these risks, individuals can make informed choices that promote long-term health.